We constantly see dramatic medical breakthroughs making the news, right? But have you ever noticed that years later, those promising new treatments almost never make it to actual pharmacy shelves? It's a massive, multi-billion-dollar question that has literally plagued the pharmaceutical industry for decades. I mean, billions of dollars are poured into research, yet the pipeline from the laboratory to the patient is, well, it's incredibly leaky. We're talking about potential cures for devastating diseases that just seem to vanish. So why exactly does this happen?
The answer really boils down to one absolutely staggering statistic. Historically, over 90% of drugs that successfully clear animal testing go on to fail in human trials. Just think about the sheer scale of that failure rate for a second. 9 out of 10 experimental medicines that look completely safe, totally effective in a mouse, a rat, or a dog, end up being toxic or just plain useless when given to a human being. It raises some massive, fundamental questions about the tests we currently rely on to develop life-saving medicines.
Welcome to The Explainer. Today we're taking a fascinating dive into a monumental shift in the future of medical science. We're looking at a revolutionary new approach being pioneered right now in the UK, an approach that aims to not only fix that 90% failure rate, but potentially phase out traditional animal testing for good.
But before we uncover how scientists are actually growing living, beating human mini organs in the lab to make this happen, do make sure to hit that like button and subscribe. It is absolutely the best way to stay on the cutting edge of science and innovation with us. All right, let's dive right in.
Section 1. The drug testing problem.
To really understand this leaky pipeline, we first have to look at the sheer scale of our current model. In the year 2025 alone, there were 2.54 million animal testing procedures conducted in Great Britain. Now, to be fair, that number is actually down about 3.8% from 2024, so there's a slow trend in the right direction, but it's still a massive figure. Over 90% of these procedures used mice, rats, fish, and birds. But crucially, about 1% still relied on specially protected species, and that includes cats, dogs, horses, and monkeys. This is essentially the bedrock of how we currently test for drug safety.
But, as we saw with that 90% failure rate, this bedrock is seriously cracking. Professor Matthias Zillbauer from the Cambridge Stem Cell Institute kind of hit the nail on the head here. The fundamental issue, it's a biological mismatch. He says, and I quote, "A lot of human diseases either do not occur in animals or occur in a different way, because they're not human. A mouse cannot tell us that." It makes sense, right? A mouse is not just a tiny human. Many complex human diseases just don't occur naturally in animals, or if they do, the underlying pathology is totally different. We're basically trying to translate mouse biology to human biology, and the science is telling us loud and clear. A mouse just cannot reliably tell us what's going to work in a human patient.
Section 2. Enter the organoids.
So what is this sci-fi sounding solution that's actively revolutionizing the pharmaceutical industry? Enter the organoids. They fall under a broader category researchers call NAMs, or New Approach Methodologies. Now, researchers have actually been growing these for over a decade, but the technology has just recently hit a major tipping point. An organoid is basically a tiny, simplified version of a human organ or tissue. We're talking about clumps of tissue that are often smaller than a single millimeter. But don't let their microscopic size fool you. These tiny structures mirror the key features of full-scale human organs. They show us how those organs falter when diseased and exactly how they respond to experimental drugs.
Notice the stark contrast here. On one side, you have the traditional mouse model, a completely different species, requiring us to basically guess how an experimental drug might translate to humans. On the other side, you have human organoids. And here's the kicker. These aren't just generic cells. They are grown directly from cells donated by actual NHS patients. This means the miniature organ sitting right there in the lab dish retains the unique, specific biological and genetic characteristics of the individual patient who is actually suffering from the disease. It is just an unparalleled leap in precision.
Section 3. The UK's £22 million solution. Now, the UK's life sciences sector is already a total powerhouse, generating around £147 billion in turnover and employing over 360,000 people. And now they're throwing some major financial weight behind this revolution with a massive £22 million government initiative. The lion's share, $20 million from the Medical Research Council, is funding a brand new preclinical translational models hub based right at the Cambridge Biomedical campus. This is geographically crucial because it sits just minutes away from major NHS and industry partners like AstraZeneca and GSK. Alongside that hub, another $2 million from Innovate UK is being injected into 9 cutting-edge, independent projects specifically aimed at finding alternatives to testing on dogs and monkeys.
The most crucial part is what that £20 million Cambridge hub is actually going to do. They aren't just running isolated experiments. They're creating a vast, shared library of standardized, validated human organoids. This library will be available to academics and the broader pharmaceutical industry. Imagine if drug developers could essentially just check out standardized human gut, brain, or tumor models from a library. They could test their drugs on them immediately, completely bypassing animal trials while getting vastly superior data.
Section 4. Inside the new tech.
Let's pull back the curtain on some of these futuristic projects, because the innovation happening right now is absolutely mind-blowing. You have companies like CN Bio building tiny working versions of human organs so small they literally fit on a standard microscope slide. This is often called body-on-a-chip tech. Then there's Vivosphere, who are literally growing beating human heart cells suspended in tiny gel spheres. Camgene Therapeutics is creating lab-grown human ear models to test hearing loss therapies, again, without using animals. And it's not just biological, it's digital too. Companies like IoTech and Nanograb are leveraging artificial intelligence to predict exactly how a drug will move through the human body, using lab data alone to replace traditional animal pharmacokinetic studies. It's wild.
So how does this whole new testing paradigm actually work in practice? First, you grow the mini organ from the patient's cells. Second, you apply the new experimental drug candidate directly to it. Third, instead of guessing based on a malice reaction, you get to observe the direct, localized human biological response. And fourth, you detect any toxic side effects incredibly early in the process. Basically, if a drug is going to fail, we find out in the dish, not in an animal, and certainly not in a human clinical trial.
Section 5. A faster, kinder future.
Ultimately, this technology points to a massive dual victory for both human health and animal welfare. While researchers will eventually grow a full range of organoids, from beating heart tissue to electrically active brain cells, they're anchoring this science to very real, urgent human conditions right now. Professor Zilbauer's team, for instance, is starting with organoids specifically for inflammatory bowel diseases like Crohn's and ulcerative colitis. Other teams are focusing immediately on growing specific human tumors to radically speed up the discovery of effective cancer treatments without needing that animal middleman.
I really want to highlight this visionary quote from Dr. Juliet Dukes from the charity Replacing Animal Research. She points out that unlike animal models, these organoids offer the promise of truly personalized medicine. Because we can grow an organoid from your specific cells, doctors could theoretically test multiple different drugs on your mini organ in the lab, find out exactly which one works best for your unique biology, and then prescribe you the perfect bespoke treatment. It's a complete game changer in how we think about healthcare.
We've seen how global regulators, including the UK's MHRA, are already committing to reviewing this non-animal data. The massive financial investments have been made, and the technology is no longer science fiction. It is living, beating, and reacting in laboratories today. So I'll leave you with this to ponder. As we stand on the precipice of personalized organoid-based medicine, are we finally witnessing the beginning of the end of animal testing? Thank you so much for exploring the cutting edge with us here on The Explainer. Catch you next time.